Now we're going to study Major Histocompatibility Complex or HMC, MHC, sorry. Major Histocompatibility Complex. What is it? Major Histocompatibility Complex is a special protein present on the T cell. It is a T cell receptor, that is TCR. On the T cells, these major histocompatibility complex are present, which is helpful for the identification of antigen. It is nothing but a connection between antigen and tissue. Major histocompatibility complex helps in the connection between antigen and the tissue. Firstly, what is the function? The MHC function is helps in the recognizing antigen towards the T cells. T cells don't know what kind of antigen is present and how to recognize this, but MHC are the receptors TCR present on the T cells, which help in the recognizing antigens towards the T cells. So the MHC in the human body is known as HLA. That is, it's a human leukocyte antigen is nothing but a TCR. This HLA gene is present on the short arm of chromosome 6. So there are three classes of MHC. First is class 1, class 2, class 3. So this is the class 1, and this position is antigen binding site. So class 1 and class 2 are transmembrane protein molecules, that is transmembrane glycoproteins. If this is a cell membrane, this class 1 and class 2 are present in the between it, above and the below also. Like in between the transmembrane, this class 1 and class 2 MHC are present. Coming to the structure of class 1 MHC, class 1 major histocompatibility complex contain two subunit, alpha and beta, and alpha subunits is of alpha 1, alpha 2 and alpha 3. The molecular weight of whole alpha molecules is 45,000 daltons, while beta subunits is 12,000 daltons. See, these are the alpha subunits, and this is the beta subunit. The weight of alpha subunits is 45,000 daltons, while beta subunit is 12,000 daltons. So class 1 major histocompatibility complex are present. They are found in almost all nucleated cells. So it is helpful for the binding of antigen. And the next is class 2 structure. Class 2 MHC is associated with leukocytes. These are associated with all nucleated cells, while class 2 MHC is associated with leukocytes, which are involved in cell mediated immunity. That is, which leukocytes involved in the cell mediated immunity? They are macrophages, dendrite cells, NK cells. So class 2 major histocompatibility complexes are associated with dendrites, macrophages and NK cells. Class 2 MHC is also a transmembrane glycoprotein and contains two subunits, alpha and beta, of course. Alpha is of alpha 1 and alpha 2, and beta subunits are beta 1 and beta 2. The weight of alpha subunits are 30,000, while the weight of beta subunits are 28,000. Here we can see this is alpha 1, alpha 2 and beta 1, beta 2. While here alpha 1, alpha 2, alpha 3 and the beta. So alpha 1, alpha 2, in class 1, alpha 1, alpha 2 are responsible for antigen binding site, and in class 2, beta 1 and alpha 1 are responsible for the antigen binding site. In the class 3 is not like a class 1 and class 2, but these are the produced by proteins. Class 3 MHC are secreted by the proteins involved in the immune functions. That is, in the complementary system, the cells or the proteins involved in the immune function or the complementary system produce this class 3 MHC. So some are C2, C4, TNT, these cells involved. In the class 2, with the help of CD4 antigens, class 2 MHC represents antigen towards T cell, while class 1 with the help of CD8 helps represent antigen towards T cell. See, the class 1 MHC with the help of CD8 helps antigen representing towards T cell, while class 2 helps with the help of CD4 only. It helps in binding. The class 1 MHC helpful in representing antigen towards T cell with the help of CD8, while class 2 MHC with the help of CD4 cell represents antigen towards T cell. Now coming to the pathway. Pathway is nothing but how it helps in representing the antigen towards the T cell. Firstly, see class 1 MHC pathway. It involves 4 steps: antigen uptake, antigen processing, peptide MHC association, and the fourth is expression on cell surface. Antigen uptake, processing, and peptide MHC association and expression on the cell surface. Firstly, antigen uptake. Antigen uptake is nothing but uptaking the antigen. This is the first step. And the second step is the uptaken antigen contains many complex structure, but from this antigen proteosomes are produced. And from that peptides, from these proteosomes peptides are formed. These formed peptides are bind with MHC. This is nothing but peptide MHC association. Then after when peptide binds with MHC, these MHC are taken up onto the cell surface, and with the help of CD8 cells, antigen is represented to the T cell. So this is the pathway of class 1. Antigen is uptaken from the outside of the cell. Antigen is uptaken. Since antigen is of complex in structure, we process the antigen. Antigen processing takes place, and from that proteosomes are produced, from which peptides are formed, and these peptides bind to the MHC. MHC are transmembrane glycoproteins, no? So these peptides and MHC association takes place, and this MHC peptide association complex is taken up onto the cell surface, which with the help of CD8 it helps in binding, it helps in representing antigen towards the T cell. In the same way, class 2 pathway, it is also a 4 steps pathway, but here the antigen is extracellular. But in the processing of antigen, endosomes are required. So in the class 2 MHC, an invariant chain is present in inactive form, and to turn into active form, this invariant chain should be dissolved or digested. So these endosomes also help in digesting the invariant chain. Also these endosomes process the antigen to produce proteosomes. So when invariant chain is converted to active form, the peptides are bind to MHC, and this is taken up to the cell surface. See, while this is the intracellular antigen, here in the class 2 pathway, it is an extracellular antigen and taken up into the inside of the cell. Here the processing takes place with the help of endosome. It also helps remove the invariant chain of MHC, and the peptide and MHC association takes place, which is taken up onto the surface of the cell. Here with the help of CD4, this class 2 MHC represents antigen towards T cell. This is the pathway of MHC. So the function of MHC. Firstly, the most foremost function is MHC, major histocompatibility complex, represents antigen towards T cell. And MHC reacts both for self antigen and non-self antigen also. It also helpful in order to mediate certain autoimmune diseases. It will have a defense action against disease and infection. It is also responsible for the individual smell of the people. So the functions of MHC are, firstly, it represents antigen to the T cell. And the next is, it is responsible for the individual smell of the people. It also helpful in mediating certain autoimmune diseases. Also have defense. It have defense action against diseases and infection. And major histocompatibility complex reacts both to self antigens and also non-self antigens. So these are the functions. Firstly, representation of antigen towards T cell, and nextly react both to self and non-self antigen and helpful to mediate autoimmune disorders, and have defense action. Most is defense action against disease and infection, also responsible for the individual smell of the people. So this is about the major histocompatibility complex. Class 1, class 2, class 3, and class 1, class 2 are transmembrane glycoproteins, where class 3 is secreted by the proteins helpful for the cell mediated proteins involved in the immune functions. So this function is nothing but helpful for the representation of antigen towards T cells. And human MHC present in the humans are HLA, human leukocyte antigen. This gene is present on the short arm of the chromosome 6. And the class 1 MHC contains two subunits, alpha 1, alpha 2, alpha 3 and beta. The molecular weight of alpha subunits are 45,000 while beta is 12,000 Daltons. And in class 2, beta 1, beta 2 and alpha 1, alpha 2 subunits are there. The molecular weight of alpha 1 and alpha subunits are 30,000 Daltons, 33,000 Daltons, while beta subunits are 28,000 Daltons. Here in class 2, beta 1 and alpha 1 is responsible for the binding of antigen, and in class 1, alpha 1 alpha 2 is responsible for binding with the antigen. And this is the pathway. The pathway mainly involves 4 steps: antigen uptake, antigen processing, peptide binding, peptide MHC binding, and uptake of the complex to the cell surface of the cell. Both class 1 and class 2 pathway is same, but in the class 2 endosomes are present. Endosomes help in the processing the antigens, but also it helps in removing invariant chain present on the class 2 MHC. And in class 2 pathway, the antigen present is extracellular antigen. So this is about the antigen. Function is nothing but representation of antigen towards T cell, and both MHC reacts to both self antigen and non-self antigen. It also helpful in mediation of autoimmune disease. It have defense action against disease and infection, responsible for the individual smell of the people. Thank you.